Aloha and welcome to our video on the cell cycle and mitosis. The goals of today's video are to identify the phases of the cell cycle, to discuss the activities of each phase of mitosis, and then finally we'll compare reproduction in eukaryotic and prokaryotic cells. Okay, so here is our simple model of the cell cycle. It starts right here when we have a cell that's formed, one of the daughter cells. So it first goes into this G1 phase, and that's G1 for growth. And what it's basically going to do in this one is it's going to get to be a full-size cell. So when they're first formed, they've just split. They're going to be a little bit smaller, so they're going to have to increase in size, and that'll be making a few new organelles, and it starts doing its normal functioning processes. So if it's a muscle cell, this is when it'll act as a muscle cell. If it's going to be a skin cell, this is when it's going to act like a skin cell and do what skin cells do. After the G1 phase, it enters the S phase here, and during the S phase is when it goes through DNA replication. And this is important because before the cell can split, it has to copy its DNA so that both of the daughter cells are going to get their own copy of DNA, and they can then function as a working cell. After the S phase, it goes into the G2 phase, and this is going to be the preparation for mitosis. So it might get a little bit bigger. It's going to start making sure it has enough copies of the organelles so that both cells, both daughter cells, will have a copy of the organelles. And then it leaves the normal working phase, which we call interphase. And this is where the cell spends most of its time. And then it goes into this last part over here, which we call cell division. And cell division is sometimes called the M phase, and that's for mitosis. And we'll go through the steps of mitosis here in just a few minutes. But mitosis is when we divide the nucleus, and then ultimately through cytokinesis when we actually cleave and split the cell into two. So that's a quick overview of our cell cycle. First it goes to the G1 phase, then it goes to the S phase, then it goes to the G2, and then it goes into this M phase here finally. And at the end of the M phase, it'll split and it'll form two daughter cells, and each of the daughter cells then starts again here in the G1 phase. So again, just in review, we're going to talk about interphase. Remember, interphase was made up of the G1, the S, and the G2 phase, and during interphase, the cell is going to grow. It's going to make copies of its DNA and other organelles, including the centrioles, and this is when it gets ready to divide, but it's also when the cell is functioning as it's supposed to. Once the cell has got ready for division, it enters in the M phase and it starts mitosis. The first stage of mitosis is prophase here, and what you'll see is a couple different things happening. You'll see that the nuclear membrane here is starting to break apart. All right, so now the nucleus is starting, the membrane's dissolving away, and that'll expose out our chromatids and our chromosomes. And this is our genetic material. So you can see these right here. And what they're basically doing is they're thickening up. They're bundling themselves up and winding themselves tighter and tighter, and that keeps them together. When the cell's in interphase, that DNA material is going to be all spread out and wispy and kind of a tangled knot. But what it'll do when it's time to move and it's time to split the cell is it'll bunch up together, and that makes it a little easier to keep track of where everything is. You'll also notice up here we have our centrioles, and our centrioles play a part in the division of the cell. And they're going to start forming what we call this spindle in between them, these spindle fibers. And that's what will actually pull the chromosomes to the opposite sides of the cell or to their poles. And that takes us through prophase. During metaphase, and it starts with the M, so we're going to work about the middle or the metaphase plate, what we'll notice is that the chromosomes here are lining up in the middle of the cell. And the reason this happens is that just kind of makes it real simple to make sure that both sides get what they need. Okay, and that's the important thing, is we want to make sure that when the cell divides, each side of the division results in a daughter cell that's going to actually have all the pieces and parts it needs to be successful. So you can see that the centrioles that started off on one side have migrated to the opposite poles. The spindle fibers have now stretched across the cell, and you can see those coming across this way here. Those are the green lines. And this is metaphase. Remember, metaphase is the middle. All of the chromosomes line up here on the middle. Next up comes anaphase, and in anaphase we notice the importance of the spindle fibers. They attach to the centromere of the chromosomes, and they're going to pull apart. Notice this once was a nice little X-shaped one in here, and its centromere is here, and it's going to pull these apart, and we end up with a half one here and a half one there, and that's a complete set. These were once pairs. So by doing this over and over again is how we split the cell's DNA, And notice it's moving to the opposite sides. And we start to see a little bit of a cleavage furrow here where the cell's starting to look like it's going to start pinching in together and start moving. So anaphase is when we start separating and moving the chromosomes to the other sides of the cell or to their poles. Now, the fourth and final phase of mitosis is what we call telophase. In telophase, we'll notice that the spindle fibers have all disappeared at this point in time. We have centromeres that have located to opposite ends of the cells, okay, and we're starting to see this reformation of a nuclear membrane. So it's forming a new nucleus. We'll also start to see this DNA start to unravel so that it can be used. And we'll talk about the using of DNA in a couple units. You'll also notice that this cleavage furrow, this division, is starting to get a little bit more pronounced here, and that'll take us to our last and final phase, the M phase. Okay, the last stage is cytokinesis, and cytokinesis is when the cytoplasm splits and we form our two daughter cells. And what you can notice in each of the daughter cells is that the nuclear membrane is completely done, and we have our two new nuclei, and you can see that the DNA is starting to squiggle out again, and this is how it becomes useful. And these cells will now enter into interphase, and they'll start growing to become a regular sized cell and doing what they need to do, and ultimately they'll get ready to go through the process of mitosis again. So as a quick review of mitosis, we start off here in prophase. And in prophase, remember we had our centromeres and our spindle starting here, the nuclear membrane's going away, and our chromosomes are starting to thicken up here a little bit, and they're getting ready to be moved. The next stage is metaphase, and that's where they all line up here on the middle, and our spindle fibers start attaching to the centromeres of the chromosomes. During anaphase over here is when we see the separation of the sister chromatids, and they'll start migrating to the poles. They're pulled there along that spindle fiber by the centromeres. Then we enter here into telophase, and in telophase we see the disappearance of the mitotic spindle. You'll also start seeing the nuclear membrane starting to form, and we're starting to form our two different nuclei. You'll also notice that we're starting this whole cytokinesis process, which finishes up over here, and that results in our two cells. And then our daughter cell will then go into interphase, where it'll get ready for the whole process to start all over again. So one more time as a quick review, our new daughter cell is formed, and it goes into this G1 phase where it performs its typical activities, and it'll grow to be a normal size. Then it goes into the S phase where it'll copy its DNA. Now it can stay in this G1 phase for an extended period of time, or it can be a relatively short amount of time, depends on the cell, and the lessons will go through that with you. After the S phase, it goes into this G2 where it makes a copy of its organelles and it doubles in size almost. Then it enters into mitosis, okay? So interphase is where it's doing its normal activities, it's copying its DNA, it's copying the organelles, and then it goes into mitosis where we have our prophase first, where the nuclear envelope will break down and we start forming the spindle fibers. Then we go into metaphase, where the spindle fibers line up the chromatids in the middle of the cell. Then we go into anaphase, where the sister chromatids are going to separate at their centromeres, and the spindle fibers move these separated chromosomes to opposite sides of the cell. And then we enter into telophase, where the nuclear envelope starts to form. So it forms a new nucleus, the chromosomes start spreading out, all of that stuff gets ready. Cytokinesis is also going to be going on here, and we'll break into our two daughter cells, and that takes us right back here to the G1 phase. So mitosis is how eukaryotic cells divide. We also have the prokaryotic cells that don't have a nucleus, so there isn't this big process of mitosis going on. Instead, they do a process called binary fission. And binary fission quite simply means is we start off with our cell, and we have our plasma membrane. Remember, they only have the membrane at the very end outside that separates the cell from the world. There's no internal membranes, and their DNA is just floating inside the cytoplasm. Now, the DNA will duplicate itself, and then the cell will divide, and then we have two daughter cells. And these are going to be carbon copies of themselves. And that's why it's done a little bit quicker, is because there's not the long process that it goes through to do this. It'll just duplicate the DNA, and then it'll just divide that way. So that's it for our video. As always, the lessons will go into it a little bit more in detail and depth. And hopefully you'll do well on the lessons, and we will see you again next time.